get this shared. So this is a pretty bold statement, but as I mentioned, I've worked in a lab before, and losing samples in freezers is unfortunately more frequent than it should be, especially when we go to the lengths that we do to receive, process, generate these samples. Sometimes they can never really be regained if you lose them. So I'd like to kind of talk a little bit about some of the vulnerabilities in that sample life cycle that Sample Manager can help with. So of course, upon sample receipt or creation, if you are getting a sample into the lab and folks aren't accessioning or marking that sample down as received, it can easily get lost. You also may not be capturing the type of metadata that you need to be able to come back to that sample in the future as well. And so making sure that everything is within kind of that right context during sample labeling, as well as metadata capture is really important. When you add it to storage, so I think both Dylan and Sydney made some good points around having to create workarounds in their lab, even with such a flexible system like ours that can allow for the sample to potentially get lost along the way. So ensuring that you've got a good audit trail, even with those workarounds, is going to be really important. Retrieving them from storage. So maybe somebody adds them into the lab. I think Dylan talked about sort of attrition in that academic space. I've definitely experienced that, where a PhD candidate graduates and leaves, and we're kind of left with an entire shelf of unmarked, unlabeled samples that we have no idea what to do with, but we're too afraid to discard them. So being able to kind of go get those samples and know exactly where they are and what they are, and maybe potentially what they've been used for in the past is really important. And then kind of lastly, like as you're rounding out experimental data or preparing reports for INDs or IRB submissions, if your experimental data isn't capturing all of that metadata about the samples, it's very easy to then kind of lose that sample's context and not have those be usable in the future. So as I already showed you, Sample Manager is mainly a sample tracking tool. We've built all of these other components around it to really make it a very holistic laboratory system that allows you to track a lot of other things. So I'm actually going to hop into a live demo and show you some of these components real time as we're going. And again, I'll be kind of my plug for please ask any questions in the chat. Because this is going to be a demo, I'll try and sort of look at that more frequently to answer them sort of as I'm maybe in that area of the product. So this is our fake LabKey lab, which we as administrators have completely customized. So I've created the 7 different sample types here. I've named those sample types. I have completely decided what specific fields should go with blood versus CSF versus DNA, because all of those things obviously can be very different. We can also see a breakdown of the sample statuses within the lab as well. So very quickly, I see I have a lot of DNA samples registered, but most of them have already been shipped out maybe to a collaborator. I can get a quick understanding of the notebooks maybe that I'm working on, or things that I need, I'm waiting on review, or if I have a review to do, that would show up here as well. I can see what freezers we have in the lab, as well as a very quick understanding of the amount of capacity that I have available. So I think Sydney kind of touched on this, of making sure that you know, A, when you might need to start consolidating, or B, when you might have to buy a new freezer, for example. This kind of gives you a heads up along the way. And lastly, on this page, I get an understanding of any workflow jobs that I have on the list. So I've got a couple of user conference jobs that we'll get into later. So because this is really about like how never to lose a sample again. If I were to come into this system and want to register some more samples, I'm going to go into the sample type that I have. In this case, we've got blood sample type, which again is something that I configured. I'm capturing all of this metadata, customizable by me, in the system, as well as getting sort of a unique barcode, and the ability to track an external barcode. So if I'm receiving samples that have already been barcoded, or I've got RFID barcodes in my tubes, I can use that feature as well. When I'm ready to add samples, I can do that very easily, either manually, or if I've got a file import. Oftentimes we've got manifests coming to us from other folks, or it's been generated out in the field or other places. And so I'm able to, do a bulk import if I need to, or I can sort of add these singly. When samples are added, you can see that we have a unique sample ID associated with these samples. In my case, I have customized my blood samples to just be named blood with sort of a unique number at the end of it. You can get very advanced and very creative with this naming pattern to really make a human readable, unique ID that is pertinent to your lab. If I look at an individual sample, I can get a lot more information about it than what's just on the grid. So I can see that this blood sample has been discarded, but I've got 3 aliquots that were created for totaling 3 mils. I can also see that this blood sample has been referred to in one of my notebooks. And so these are all sort of blue live links to the data that's associated with it. We'll kind of play around with that in a minute. I can also see all of that customized information that I want to capture on my blood samples here. So I can see, what project it's part of, when it was drawn, and when it was received. And also kind of that giving that context of the blood sample. So in this case, this blood sample came from mouse 11 and was part of study 1. I can also see this in our lineage graph that allows me to see kind of those inputs of my blood sample as well as some of those outputs. So we already know that there are 3 aliquots from this blood sample that have been created, but we can also visualize that a PBMC sample was generated from this blood sample. If I want to do something with those aliquots, if I need to ship them out or take action on them or assign them to some workflow jobs, I would be able to do that here. I can also visualize any assay data that's been run on this sample or its aliquot. So in this case, this blood sample has undergone a CBC assay, and my results are here, as well as if it's undergone any workflow jobs. So I can see that, again, sort of my user conference workflow job is here and kind of partially complete at this point. And then probably my favorite thing about what we do in Sample Manager is providing this sample-centric timeline view. And so this is really a good way to not lose samples ever again, right? So I was able to uniquely register the sample, put it in the context of where this blood sample came from. This wasn't just some random blood sample that kind of fell out of the sky, right? It was from that mouse, from that study. And all of the things that have happened to the sample since I've registered it into the lab. So when the sample was registered and added to storage, what assay data it was associated with, what workflow jobs it was part of, when it was discarded. And so at any of these points, I can actually look at these events and get more details about the event itself and find out even more information if I'm going back and looking for, like, what is this sample really? What has happened to it? We also provide the ability to export this audit log. So if you have auditing needs where you need to kind of do this, or if you're filing a discrepancy or trying to capture some change, you would be able to, export this for easy record keeping later as well. So I'll go back to this overview, and this sample has been discarded from storage. But if I look at the aliquots that were created, I can see the grid and see that these samples are within the system. So if I click on this aliquot, my blood sample aliquot here is in my -80, in this shelf, in this rack, in this box, in D5. So if I go into storage, one of the main things that I want to be able to do when I'm looking at a sample in storage is identify it from the other vials in the system. And so in this case, we can very easily see that our blood samples are here in red, as well as we've got some plasma samples and some serum in this box. Any samples that have been checked out, we're going to hold that location for those checked out samples so they can come back, kind of saving you that freezer space over time, as well as whatever organizational choices you've made to get samples into boxes. So in this sense, we don't have probably a very good organizational scheme with plasma and serum and blood being all in the same box, but you can imagine that that would probably reflect very differently in a real lab. So I see maybe someone's trying to answer a question, so feel free or ask a question. Continue to just kind of type that in the Q&A, and I will continue to go on here until I see that question. So as I mentioned, kind of all the things that you can do within that you're doing within sort of the physical space, you can do within this digital space. So if I'm interested in checking out a sample, I can do that individually. I can do that kind of together and check samples out for my demo. As I mentioned, when they're checked out, those spaces are reserved for these blood samples. So when I've done my work and have come back in, I can also choose to check these in. Upon check-in, we do a really cool thing where we're actually decrementing the amount of the sample that you used during thaw. So in a lot of cases, this is probably really unlikely to happen with a blood sample, but you can imagine for serum or plasma, if you have not created single-use aliquots, you check them out for a big Luminex assay or something like that, you used 50 microliters across the board. And so now I don't have to go in and update every single amount of sample that is left, but just the amount that I used upon checkout. Of course, we're also providing that increment for a freeze thaw count. In the case where you've made a mistake, you did not actually check these samples out, you can turn this off. And this is really unique from other systems because I've used other systems before, and it's not very helpful if you end up getting kind of misaligned with what reality is. And so we're trying again to give you all of these different mechanisms to reflect what's actually happened in the lab. So I'll say for my assay. And when I check these samples in, I can actually look at them individually and see, that sort amount has been decremented, my freeze thaw count has been incremented. The other really awesome thing, so we talked about sort of sample retrieval. So if I'm going into my freezer and I see this box, but my digital storage says that there is a blood sample here, and my physical storage does not reflect that, I'm able to look at the individual location history of this D5 cell and see all of the different things that have happened in the cell over time. And so I might come in and say, oh, wow, somebody checked out this sample, in which case it was me. Maybe I need to go check with them. Maybe they forgot to actually place it back in the freezer, even though they sort of checked it in digitally. Okay, see a question here. Can we add a column to log different sample volumes for batch upload of samples, or do we have to edit each sample individually? So yeah, that's a great question. What we currently support right now is sort of in bulk. You can decrement or update those samples individually via this mechanism. You can do it individually, of course, but the other way that I might recommend doing this is using a file upload. So I mentioned that kind of at the beginning with sample registration. You can also use a file to update samples within the system as well. So in that case, where it might be very granular, very different between samples, you would be able to make a bulk update at kind of in a single go at that point as well. Some other things to kind of note here. So if I'm examining my box and wondering exactly the context of these samples, I'm able to look through. We've got our grid view of the samples below here. I can actually select a few samples. We'll kind of do this and choose to view, the selected samples. And in that case, now I've got a very refined view of my samples. If I am looking for more information about the blood samples within this box, I can see those samples. I can also see, again, that context, where the sample came from, what project it might be in, what the barcode is, if I've got my scanner in front of me, things like that. Can you use Sample Finder to filter data based on assay data? So this is actually a great question. I'm going to come to Sample Finder in 1 minute, and so I'll kind of loop back to that one as well. Okay, so the other kind of things that I want to highlight here in this freezer management is what I mentioned before about the forced creation of workarounds, or creating using a system that isn't completely flexible in how you set up your freezers. So I'm able to manage my own storage units and create really any type of storage unit that I'm interested in. So if I saw that I needed a different size box, you can see on our demo site, we've configured this very heavily. I can go through and create a new box size and structure as I need to. At any of these given places, we also give you the options for how these boxes are displayed and how the cells are displayed, because we do understand that different freezers or different types of boxes may have a different display on them. I'll open up my -82 to show you kind of the configuration. So this is, again, completely configured by me or an administrator in the lab that has said, we have a shelf inside this freezer that has a rack that has 2 boxes. We have lots of folks kind of using this to create whatever their freezers look like. And you can see kind of in my 4th shelf here, I've got sort of a grab bag shelf, which has some random plate and a bag of samples in it. Somebody asked the question that they have materials in kind of funny containers. And so hopefully that kind of just demonstrated to you that you can really create your own structured containers kind of as you'd like within the system as well. So other things that I can keep track of, I think this was coming back to maybe a question in my previous demo or my previous talk, as well as sort of in here, is this ability to find samples later. So if I am unsure of all of the different samples that I have from a given mouse, for example, maybe we'll go in to mouse 11, which is the mouse that I associated with the samples earlier. I can actually go into my source type, which again, totally configurable by me. I've created a source type called mice, and then registered my individual mice to kind of be captured here. I can see all of the different samples and aliquots that have been generated from this mouse. I can view that via lineage, which gets a little bit messy, frankly, when I've got a lot of samples generated. Or I can view it kind of in a grid view. So there's 71 samples from this mouse. And so if I found out maybe in sort of a clinical trial case where I've got a subject that has withdrawn consent or something like that, this provides me with a really quick view of what are all the samples that have ever been generated from that subject, where are they, and I can do kind of a bulk discard or things like that. In this case, I can also sort of be more creative and say, okay, well, I'm looking for samples that have come from this mouse, and I want to do things with it. So I can select my blood samples, I can start aliquoting or adding them to pick lists or adding assay data, things like that. But let's say that I'm actually interested in all of my samples that have come from, let's see what gender, this is a female bulb seed mice has had. In that sense, I would use Sample Finder, and somebody was asking about this in the last talk. So I will look for mice that have come, that female mice that has, that are bulb C. And I can actually look for all of the samples that have ever been generated by these female bulb C mice, which gives me 720. I can then sort of interrogate this further. So if I'm looking for blood samples that have come from female bulb C mice that are over a given amount, I would be able to sort of create a filter and say, I want, at least 0.5 ml of blood here. And that then narrows my search down to 37 samples. I can save this. I can decide to export this for kind of a report or analysis. I can select all these, add them to a workflow job, assign that work to someone else. I kind of have a lot of things at my disposal. Something that we are working on currently that will be out soon is actually the ability to find samples based on assay data in the system as well. So just like I have cards for source and parents, we're going to have an assay card that allows me to find samples based on assay data. So that might be find me all samples that were negative for mycoplasma or something like that, or find me all samples that came up with a positive maybe influenza result in Dylan's case. So really kind of gaining that flexibility of querying within the system. Okay, so kind of the last thing I think I want to touch on here is, now that I have these samples uniquely registered, I know exactly where they are in the freezer. I'm able to customize that freezer to really make sure that that digital space is matching the physical space. Maybe I want to perform some work on this sample and capture that in my ELN. So I would have the ability in a notebook here. I've got sort of my demo PBMC isolation. I created, excuse me, this notebook from a template. So the only thing that I've really modified from this template is adding my sample IDs at this point. I want to show what this data connectedness means. So we both highlighted the fact that these blood samples are different. It's not just regular text within the system, but I also can hover over it and get sort of my direct link to that blood sample. So if I want to then click on that blood sample, I would be able to do that, find out more information about it. Maybe I realized kind of halfway through, shoot, I don't have enough sample. Are there other aliquots available to me to kind of pull into this experiment? I would be able to do that as well. I can also see very quickly that this blood sample was part of a notebook, so I can go back to that notebook and continue on with my work. The other thing that will happen here is, as I'm exporting or kind of snapshotting this data after final review, the notebook as well as the data associated with the notebook will all be exported in sort of this finalized format for me. And then lastly, I'll kind of mention this co-authorship. So Bernie and I, or Andrew and I here are co-authors on this one. So really reflecting that sort of physical reality of most of us are collaborating within the lab. Not a single person is really doing their own experiment at a given time. So allow us to co-author these notebooks in line with how we're doing the experiments in real life.